All right, we're going to kick off the next panel, Enhancing Cannabis Testing Standards, Accreditation, Methodology, and Regulatory Reforms, moderated by Sherman Hom. You're on.
Good morning. I am Dr. Sherman Hom, Director of Regulatory Affairs with Medicinal Genomics. The panelists will discuss issues related to enhancing cannabis testing standards. These are the scientists making sure your weed meal doesn't come with a side of pathogenic aspergillus. Okay, the panelists. First we have Dr. Darryll Barkhouse, has extensive experience in quality and R&D functions. Darryll has managed laboratories in the fields of beverage, pharmaceutical, and third-party testing. Before opening operations at the new bioMérieux Genomics Discovery Center, he directed their A2LA certified ISO IEC 17025 laboratory. Darryll developed several qPCR assays used to ensure quality and safety of food, beverage, and cannabis products. He champions testing method standardization across industries, especially the improvement, standardization, and confirmation methods. Next, we have Rafael Bombonato. He was in panel 1, has extensive quality and compliance experience in regulated industries, including cannabis, pharmaceuticals, and nutraceuticals. As Senior Director of Quality Assurance at Curaleaf, he leads quality compliance operations, overseeing electronic quality management systems, efficiency initiatives, and good manufacturing practice compliance across multiple sites. Rafael designed and implemented the quality management system that earned a safe quality food level 2 current good manufacturing practice certification. Morgan Keefer is a A2LA Senior Accreditation Officer working with Cannabis Testing Laboratories. Morgan has been nominated as an emerging professional with ASTM D37 Cannabis Committee and serves at the Institute of Cannabis Science Board of Directors. She is partnering with the National Industrial Hemp Council for hemp testing, the F344 Cannabis and Hemp Testing Scope Selection Form to specify accredited matrices in cannabis testing. Then we have Heather Krug as Regulatory Programs Branch Chief for the Colorado Department of Public Health and Environment Public Health Laboratory. 4 programs include the Cannabis Sciences Program, which has 3 sections: Cannabis Laboratory Certification, Cannabis Reference Laboratory, and Marijuana Health Monitoring and Research. Using data and information from the Cannabis Sciences Program, Heather advises on state cannabis policy and regulatory decisions. She serves as co-chair of the Cannabis Regulators Association Lab Testing and Product Safety Committee. Lastly, we have Chris Marsh, the Chief Scientific Officer of Smithers Cannabis Testing Services Division and State Director for Smithers, New York location. Chris has extensive experience in cannabis testing industry operations in 11 U.S. states and is involved in the American Council of Independent Laboratories, Cannabis Science Section, National Cannabis Industry Association's Scientific Advisory Committee, and ASTM's D37 Cannabis Committee. Okay, so the topic of this particular panel, using a question and answer format, panelists will focus on cannabis testing challenges such as accreditation, standards, allowable methods, and batch size regulations. Each will discuss ways to strengthen testing accuracy, ensure quality control, and improve consumer safety. The dialogue will include reforms such as implementing limits of detection in microbial assays, refining accreditation and proficiency testing standards, and harmonizing sample collection processes across jurisdictions. So let's see. First question is for Chris. How do varying batch size requirements across states contribute to inconsistent testing results?
Thanks, Sherman. Good morning, everybody. So batch size, I think it's probably easiest to talk about this in the context of flour, although it can apply to all matrices. But I think I've seen 50-pound limits, I've seen 20-pound limits, I've seen 10-pound limits, and I think the question is about how can batch size variability contribute to inconsistent testing results. And I think it's not hard to see how if you tested a 10-pound batch and a 50-pound batch, how you might, depending on how you sample that batch, you might miss something if in a much larger batch. So if you imagine a 50-pound harvest size batch, there could be hot spots, right? 99% of the time, I don't think we're concerned about this, but in the event of something like a microbial contamination, where maybe it's in the back corner of a room, it's much more likely that you will not catch that in a larger batch of, let's say, 50 pounds than you would in a 10-pound batch. But I think really the more important thing here isn't necessarily the batch size. I mean, it's a little frustrating that states, among other things, haven't standardized what the appropriate batch size should be. Like, I don't think we actually know what that size actually should be. I know the USP has made a recommendation on it, but I don't know that it's entirely appropriate for cannabis, and I think more research is needed. But I think more importantly than batch size is how you sample the batch, right? Representative sampling is so important, and depending on the state you're in, I know in Florida, for example, I think it's more specific. There's more specific requirements about how to sample the batch. But in states like Arizona, we will show up to a processor and they'll just hand us the sample. There's no requirement at all. And I think that's really where there's a large gap in what is the most appropriate way to sample cannabis flower batches, especially flower. I think if you follow a representative sampling regimen and you pull from the entire batch and actually do a statistical randomized sampling event, that capping the batch makes sense to me. What is the cap? I don't know, but I think the sampling is more important.
Thank you very much, Chris. So Heather, should standardization and batch size requirements be prioritized, and what would be the benefits of doing so?
Well, I think Chris just explained really well that it isn't necessarily the batch size. I think that's so critical, but I do think if it were standardized in some way, that it would benefit, again, sort of this overall goal of standardizing different things across different operators or different states. That may, again, lead us to an overall better place in the end. I think about multi-state operators, whether that's a lab or a grower or producer. And again, if you have the same requirement everywhere you operate, I just think it's easier to develop standard procedures that you can consistently follow versus having to adapt your processes for something different in every location that you operate. So I do think that is a real benefit of any kind of standardization. And I think batch size is perhaps an easier win. Again, I don't know that we have the perfect answer on what the right batch size is. Colorado has an approach that's a little bit unique compared to a lot of states. We do not have a batch size limit, but we have ranges of size of batches, and then the corresponding what we call test batch, so meaning the sample that you select from your overall batch to submit to a laboratory for testing, that scales in size depending upon how big your batch is. And I kind of like that approach because, again, you may grow only a few plants of a certain plant type, or you might grow whole, 3 rooms of the same plant, or you might make a small batch of concentrate or a huge batch of concentrate. And so to define one batch size that you can try to apply to everything, I think may be a disadvantage for certain producers. So I kind of like the scaled approach. I don't know if ours is perfect, but we did take a long time to consider different sampling approaches and try to ensure that the end test batch size would be as representative as possible, because again, nothing really matters if your test sample isn't representative of your overall batch, then the results don't necessarily mean as much as they should. So I think that, again, perhaps having defined batch sizes and then corresponding test samples is really important, and I think that's an easy standardization win in the end. And then again, I think having sampling guidelines, which shout out to us, I don't know that ours are perfect, but we have really extensive sampling guidelines. We developed them with stakeholders. It talks about different ways in which a batch might be stored and how to randomly select. We even have it's probably maybe a little clunky and not the most beautiful tool, but it's a spreadsheet, and we have a video and instructions on how to use the spreadsheet to identify the different random locations within your batch that you should select. Again, trying to really ensure that the entirety of the batch is selected and it really is representative of what you're sampling.
Thank you very much, Heather. Well, I really love this question. Rafael, how does the lack of consistency in who is allowed to collect samples impact testing accuracy, and what can be done to standardize the sample collection process?
Good morning. So yeah, my opinion is I don't think that who's collecting the sample is going to cause major variability in testing results. To Chris's point and Heather's point, how you're collecting the sample is what's going to dictate how accurate and representative of that batch those results are going to be. There's plenty of tools out there that cover statistical sampling. We have different industries like grains, for example. There's sampling, huge silos of grains. They're not just taking a little bit from the top, right? They're taking from different areas of that batch, and they're combining that into composite samples, statistically testing sometimes multiple samples to get what's considered to be a representative result for that particular batch. So I think that once standardizing a protocol on how these samples should be collected, regardless if it's going to be a laboratory, a producer, or a in-between party, which some states allows for companies to be a company that collects the sample, brings it to the laboratory to do that collection. As long as the protocol is standardized, I think we should be in the path of the more accurate and precise results.
Thank you very much, Rafael. Okay, Chris, what gaps in current allowable methods could be addressed to improve testing accuracy?
Oh, that's a massive question. It's hard not to talk about potency. We just had a whole session on that. I think the first thing I would say is in terms of, what's allowed, I think what I would call the fundamental processes that we do that aren't test methods are very important. So like homogenization of flour of different matrices, the requirement for homogenization is different across the entire country. Some states don't require it at all. Some states require very, very scrupulous homogenization, just talking about flour. Again, I keep talking about Arizona, but they have very specific requirements following BAM and looking at particle size of flour once it's broken down prior to actually sampling and testing. So I think homogenization of the sample is very important. Same thing for gummies. I think I spend probably half of my days talking about gummies with people. And some states require you to pulverize and homogenize the gummy down using a cryogenic process, whereas others just let you dissolve a whole gummy. That you're going to have variability if you do that. So I think some of the processes we don't talk about enough need to be standardized. I think I'd also just like to mention too, there's other factors, like for example, internal standards. I think some labs use internal standards, some don't. If internal standards are used, I think more knowledge needs to be had by the regulators looking at them to understand that, if the internal standard is off, the whole calibration, the whole analysis could be off. My colleagues and I like to call methods that use internal standard essentially a one calibration point method. Because you could have a really great calibration curve and your internal standard would be off. And now all of a sudden, all of your accuracy and all of your data is different, even though it looks correct. So I think some of these lesser known details hiding in the shadows, they don't have the spotlight on them, but they are very important.
Thank you very much, Chris. Okay, you're up next, Morgan. What challenges exist when validating or verifying methods for cannabis testing?
Thanks. Good morning, everyone. I would say one of the primary challenges specific to cannabis testing is the lack of standardized methods that are readily available right now to laboratories as just a baseline. So there are many different tests that have to be done for compliance, like ranging from, of course, cannabinoids, but then there's also moisture, which is actually an extremely variable method. There's not a standard method for detecting the moisture, and that moisture percent then impacts the final results for cannabinoids and terpenes, generally speaking. There's also testing for pesticides, which some states have a laundry list of every single pesticide imaginable, and other states have fewer. So especially for multi-state operators, this can be a challenge because in different states they're dealing with different analytes, which are being regulated, different action limits, which are being required to meet certain limits of detection. An additional challenge is that the matrices that are available in dispensaries change every day. So if you're working in a cannabis testing laboratory, one of your clients could come to you and basically, with not very much warning at all, say, hey, we have a brand new product, we're now making granola bars. And so you have to turn around and validate a method that's going to work on granola bars without having a ton of the sample to work with, without having necessarily a true value to base your validation off of. You're working with the existing certified reference materials and proficiency test samples that are available, which are not comprehensive. And specifically in this situation, a lot of times the clients and the state regulators expect a very quick turnaround time on that method validation occurring. So it's actually kind of a longer process when it comes to adding a new analyte or a new method to a scope of accreditation. Laboratory has to let their accrediting body know that they want to add a method or an analyte to their scope. We have to then turn around and request the technical assessor to review this addition, potentially observe the method, and this process in and of itself can take weeks after the validation is complete. So for clients and regulators to expect that a laboratory can turn around and have a new method or a new analyte validated in a couple days or a couple weeks can be unreasonable. And specifically for the initial laboratories, for new startup laboratories, they definitely struggle with actually getting the materials on site to do the validation, because in order to get their license, they may have to be accredited, but then they can't be accredited until they have their methods validated, but then they can't validate their methods without the materials. So it's kind of this like weird cycle where they're using not the best materials for their validation initially, and then hopefully they're redoing the validation when they actually get appropriate sample types in, but that's not the case all the time. So I hope that answers the question, but there's a ton of challenges.
Thank you very much, Morgan. Okay, Darryll, what are the advantages of moving towards the use of limits of detection in microbial detection assays for cannabis?
Hi, if you were expecting Maria, I'm filling in for her. So for those of you who know Maria, I'm her replacement today. So using LOD is really about consistency and clarity. So if you're in the lab or you're in method development and R&D, or if you're in regulatory, having LODs outlined allows you to compare apples to apples. So you can take a qPCR test, compare it to a card reader or a Petrifilm or a culture method, and make sure that you're getting consistency. And then also it can help with the disparate matrices, right? How a test performs in flower is going to be very different from concentrated resin. So if we're using LODs, we can have very clear standards to set so that we know that our tests are fit for purpose, meaning they're fit for the matrix, they're fit for the route of entry. So something that's inhaled versus eaten or applied to the skin, the risks, for those products are very different. So they should have different limits depending upon their use. And this is how USP testing for pharmaceuticals is based. It's all about route of entry. So using clear LODs will help us with that matrix and the route of entry and all those things, and give us consistency. If you're in regulatory, it makes it much easier to roll out your tests and to help audit those things and help make improvements in that area. How many people here are on the science side, and how many almost everyone, no one on operations, okay? Being a lab monkey, I always talk about science, and I know this is science. So, using LODs, creating all this consistency across the industry and across the various states is always going to help consumer safety. It's going to make sure we release less pathogens into the marketplace and have less recalls. The flip side of that is if you're using a test that's not fit for purpose, so one that's too sensitive for the application, it's killing your operations guys, because now you're trashing batches or reworking or moving them into different markets when you didn't need to. So to throw a bone to the operations guys who are always giving you pressure, this will also help them have a consistent product and allow them to release batches sometimes that would have been trashed. Thanks.
Thank you, Darryll. Okay, Chris, what factors should be considered when implementing LOD-based detection, and how can labs ensure accuracy at these lower detection thresholds?
So just kind of following up with what Darryll said, I think that, when we talk about LOD-based detection, I think we're talking really about part of the conversations around how we validate these microbial methods, especially qualitative ones. We tend to, most states have implemented for the qualitative assays, presence, absence for Aspergillus, Salmonella, S-Tech. And I don't know if you've ever tried to do this, but has anybody successfully reached out to a company and asked them for what their LOD is on these methods? It's actually kind of hard to get sometimes. And it's because we've used the phrase fit for purpose. We design these assays to be fit for purpose. So if the purpose is to detect whether they're present in 1 gram or not, that's what the AOAC is going to, for example, assess the method against. So if we move toward LOD-based detection, I think one thing that obviously will come into play is sensitivity of the method. Microbiological assay manufacturers and developers often have to trade sensitivity for time. They want the fastest method they can get that is fit for purpose. So I think implementing LOD, would allow for a much more wide range of understanding. For example, I know some of my clients really like to know when they're approaching the limit, right? If you just have a pass or fail, then it's sort of too late. If you have a range with which you can assess their products against, they're going to know a little bit sooner, okay, maybe I have an increasing Aspergillus issue or whatever the pathogen may be.
Thank you very much, Chris. Okay, now Heather, what are the current challenges with cannabis lab proficiency testing requirements, and how do they affect lab staff performance competency?
Sure. There's quite a few issues with the proficiency tests or PTs that are available in the cannabis space. And I will say they definitely can affect staff competency if you are relying on that PT as a sole mechanism of assuring lab competency. I would never advise that is what a lab does, though. That you should have, separate competency assessments and, initial demonstration of competency before a lab staff ever even starts performing a test. And you can use a PT as part of that. You can prepare an internal blind sample. So I hope that the challenges with PT are not affecting staff competency and performance, but it's certainly possible if you're not doing the right thing that, you could be using something that is flawed or perhaps just not adequate to look at staff competence. But there are a variety of challenges with the PTs available. And CANRA did publish a white paper on best practices in proficiency testing. So if you're interested, I would definitely say check that out, whether it's as the lab, so you can kind of know what you should be hoping for in a PT, or if you're a PT provider, what regulators think is best practice. But some of the issues include that a lot of times the analytes are spiked in the proficiency test sample really high, which that's great. If a lab can detect a large amount of something, I really hope they can, because there's a bunch of it there. But they're often much higher, amounts than anywhere close to the regulatory action level. So again, that sort of the cutoff level really that labs need to verify they can test at, the PTs don't challenge that, and that isn't ideal. There's issues with matrices because marijuana itself is still a federal one scheduled or controlled substance. So PT providers can only get that if they have a DEA registration, but if they have DEA registration, they can only provide it to others with DEA registration, and most cannabis labs are not registered because the DEA won't do it. So a lot of times hemp matrices or other surrogates are used, which isn't the worst thing to use hemp. It's the same plant. We've all talked about that already at this conference, but it means the analyte ratios are sometimes different, right? You're going to have more CBD generally than you're going to have THC. So it's just not as reflective as things that they're testing every day. PTs also come in limited matrix types. So flower types are available, sometimes chocolates, maybe gummies or other types. But there's how many different varieties of different product types on the market? 1 million, it feels like to me. So again, the PTs don't always challenge what labs are actually testing, and sometimes the matrix itself presents a unique testing challenge, and they don't get to explore that in the proficiency test because it's not in that challenging matrix. And then sometimes I think one of the other issues is that, right, so different states require different analytes to be tested for. Like in Colorado, we require like 100, I want to say it's 102 pesticides to be tested for. Some states require more like 50-ish. Some states somewhere in between or less. So the PT providers are trying to cater to all these different requirements. I don't think anyone is putting 102 pesticides in their PT samples. So basically what that means is just that all the things that labs are actually required to test for are not assessed by the PT. So again, if there's a problem with a particular analyte, the PT doesn't reveal that. So it's not, I don't think these are unusual challenges given the nature of this industry, but they are challenges that labs and regulators and industries who rely on labs should be aware of in terms of how valuable or how useful that PT is in assessing the full performance of the lab.
Thank you, Heather. Okay, another PT question for Rafael. How do the types of PT sample matrices used in the industry impact the accuracy and reliability of lab results?
I think Heather touched on that a little bit. A lot of times lab is forced to buy a PT sample based on what's available and not necessarily that represents the types of products that they're testing for most of their customers. For example, potency PT is typically performed in hemp flower, right? And laboratories are testing, marijuana. So there's a very different level of THC on those two different types of flowers. So how can you assess how can a laboratory assess their true competency if they're using a sample that doesn't really necessarily meet the requirements of the samples that they test every day? Similarly, very hard to find proficiency testing samples that match all the matrices that are available out there. For example, a meter dose inhaler, right? There's not a PT for that from cannabis. Some of the topicals, there's no PTs available for topical products that would cover all the necessary assays that a laboratory has to perform. So, and most of it is related to the DEA status of THC, and that causes a lot of that challenge for the laboratories, which makes it in turn really hard for a laboratory to truly assess the accuracy and reliability of their results. That's why a lot of the laboratories that I know, they do perform their own studies on the side, or they perform spike and recovery tests just to make sure that they are recovering expected levels of, whether it's contaminant or terpene or cannabinoid.
Okay, thank you very much, Rafael. Now we move on to Darryll. What improvements can be made to proficiency testing samples to ensure they reflect real-world sample challenges faced by labs?
Okay, so one way we can strengthen proficiency testing is to really reflect the challenges of real-world matrices, like everyone has talked about. We have to use materials that give you consistent results, whether it's resin or flower or edibles or the myriad, almost infinite number of products that are out there, which is no easy task, that's for sure. One gap is the lack of diversity of organisms, and that's kind of a function of wanting to have consistency and reliability, and so, but adding diversity to that will help make sure that we're covering real-world organisms. When I was doing assay development, my favorite thing was to get real-world isolates. So we work with partners in the cannabis industry, did this as well, to get real-world isolates coming from their producers, coming from their labs, coming from their grow operations. Because something that you're going to get in 2025 coming from your own operation can be characteristic a lot different than something that was put in an ampule in 1977 by the ATCC. So to make sure we're covering and not missing these things, it's important to increase diversity in what we're testing. The other thing, so I'm a little biased, I'm a PCR and qPCR assay designer. So we need to use materials from the PT producers that are consistent and give consistent, accurate results across all the different technologies. So whether it's regular plating or it's a Petrifilm or a card reader or a qPCR assay, the PT materials have to be fit for that purpose. One other thing, we use a lot of advanced bioinformatics and data, so more data to me is better. So during the reporting process, the more metadata that we can add to those results can help improve in the future. So if you can name the method, say if you're doing PCR, the CT values, if you happen to be doing sequencing, having the reads or the actual sequences, any kind of metadata we can add to it can help improve the process for the PT producers and for the regulatory agencies. So everything has come a long way. It's very challenging to produce, consistent quality products that are supposed to be representative of a very diverse industry, and everyone is doing a good job. But now there's still a good ways to go, and I hope to see this continue to evolve.
Thank you, Darryll. So now we have some suggestions for some solutions. So Morgan, what specific reforms are needed in lab accreditation processes to ensure greater consistency and quality control in cannabis testing?
Thanks. This is kind of a loaded question. So I do want to also follow up a little bit on the proficiency testing. So proficiency testing is based off 17043, which is also an international standard. And so we talk to PT providers all the time, and one of the things that they have mentioned is that it's really actually hard to work with the data when there are so many different methods in use. And, as Darryll pointed out, there's not really very many options that you can use for microbiology on PCR and plating and MPN and all of the different potential methods. So I basically just wanted to share that from the PT provider perspective, they have frustrations with basically being able to land a plane in the data or drive a truck through the data and somehow give out pass and fail results after that. As far as reforms needed in lab accreditation, there is not much that an accreditation body can do outside the scope of the 17025 assessment. So there's an international standard, it's 17025. It's not specific to cannabis in any way, shape, or form. You can use it for testing on petroleum, automotive, electrical, pretty much any laboratory that's testing anything can be accredited to 17025. It's a standard for testing and calibration laboratories. And there are some programs which are above and beyond 17025. So these programs exist such as AOAC, and AOAC has actually recently published a guidance that is specific to food, pharmaceutical, and cannabis testing, which is way more stringent than just the 17025 requirements. So when A2LA or ANAB PJ goes in and does an assessment, we can only look at what the laboratory is being accredited to. If there were states that approached accreditation bodies and asked, hey, can you assess to our state requirements and then simplify this all in one assessment, we are capable of doing that. We just need to train our assessors to do that. Another thing that we've seen, which I think is an improvement and can continue to improve, is the state regulators actually can request the assessment report from the laboratories after the assessment's complete and then follow up to make sure those corrective actions are actually being implemented. What we see on our end is essentially we go and do the assessment. If there's any citations, which for cannabis there frequently are some citations, the laboratory has to respond to those. They provide us evidence. We review the evidence and we say, okay, we're going to renew your accreditation, or we can basically request additional evidence or additional action. But then the next time that we see that laboratory is generally like 2 years out. So if the regulators are coming in on a yearly basis, they can really like dig into the corrective actions that the laboratory is supposed to be implementing, so that we're not following up 2 years later saying, what do you mean you haven't opened your consumable log in a year and a half? So I think that can be pretty important, and I think that it'll lead to better consistency and quality control once laboratories are adopting standard methods too, because it's a lot easier for assessors to assess to standardized methods than the laboratory developed and validated methods. We have to go in and essentially look through all of the validation for all of the methods on the scope. And that can be very time consuming, especially when laboratories want, and regulators as well, want the laboratories to have kind of a couple day assessment and not us to be there for a week and a half looking at every single method validation you've ever done. So I think those two things are really steps in the right direction as far as improving the consistency and quality from the data that we see from labs.
Thank you, Morgan. Okay, Rafael, you have your chance now. What are the key areas where batch size regulations need to be reformed, and how can standardized regulations benefit the industry?
So if you were if you were here in the previous panel, you saw there was, I think, pretty much good consensus between all the panelists that the batch size itself may be not as important as how the batch is sampled. I think statistical sampling for different size batches is what's really going to bring in more reliable results. There are some tools that are available that are used by multiple different industries that allow for representative sample to occur. Once that sample comes back, for example, a very large batch is going to end up having a very large sample size as well. So in the laboratory side, the ability to harmonize that sample into a consistent, well categorized, composite is also important. It goes back into having the proper particle size, the proper mixing times to homogenize that sample to then yield, reliable and reproducible results, which is more important.
Okay, Darryll, you're in the cleanup here position. How should required test action levels and allowable methods be modified to align with the latest science and ensure accurate results?
So I'm going to repeat a little bit of what I said before. So test action levels really should be pathogen specific and risk-based. The risk-based element is what helps everybody. It helps us keep the consumer safe, and it helps our logistic folks get things out the door and keep our sales numbers where they want to be. So it's obvious something like S-Tech is zero tolerance. But something like yeast and mold, we need defined action levels. And because this should be based basically on matrix, like I mentioned, intended use, so whether it's medicinal or rec, and then the mode of consumption, because how you consume the product has very different risks. So again, those things all should be taken into account for action limits. The next is really the methods that we use, right? We need to start allowing more advanced methods. There are still states that approve only culture methods, so plating. This is one of my pet peeves as a 17025 director, is that we're constantly taking 1880s technology and have it trump 2010 technology, which makes no sense to me whatsoever. I consistently see absent results using USDA BAM or MLG, and then when I sequence them, they're absolutely salmonella, like 75% of the time. So we need to start having more advanced technologies approved so that we're really keeping that consumer confidence, consumer quality, and safety up. The last is same kind of thing as harmonization, right? We all know why things are so disjointed in the industry, and it's the federal, versus state. Hopefully that changes soon. But so the need for harmonization is really of utmost importance. Hopefully in this industry, we can do it a lot quicker than, say, pharmaceutical, because my entire career I've been hearing about harmonization and it's moved very, very slowly. But I think the demographic of this industry can make things move a lot quicker. So I'm really excited to see what happens for harmonization. Thank you.
Thank you, Darryll. Now we're going to finish up with what I call the who can land the plane the fastest. So this is the 60-second challenge. Okay, we're going to start with Rafael. If you could standardize one cannabis testing requirement, what would it be and how would you accomplish it?
That's a very hard question. There's so many of them. I think that a easy start would be micro pathogenic testing. I've seen different requirements in different states. Ranging from presence absence to less than 1 CFU per gram. To Chris's point, how would you go about testing your LOD with the 1 CFU? It gets challenging. So I think that maybe using what's available already in the food industry, having a minimal sample size where you can do a presence absence for pathogens is probably the easiest and one that's going to have the greatest impact on consumer safety, in my opinion.
Thank you. So the next is Morgan.
Okay. I would say that one change that could be made is taking into account the measurement uncertainty of results. So for those who may not be familiar, every data point, every measurement, every time you use a pipette or a balance or anything, you're adding a little bit of uncertainty to your final result. And the final result is very important when a decision rule is in place, such as like a pass-fail for contaminants. So if a decision rule is, let's say, like 20% and the action limit's at 100, a laboratory could really have a result anywhere from 80 to 115, and depending on where the result is, that entire batch is going to pass or fail. So one laboratory that you send it to, even using the same exact method, could get 101, and the other one could get 98. Now, the laboratory that gets 98, congratulations, that product's in the market. The lab that gets 101, that entire batch is destroyed. So I think it would be beneficial and value added for regulators or consumers, just manufacturers, to consider or potentially report a window of results that includes the uncertainty bands, which would really cut down on the potential for false passes and false failures for products that end up in the marketplace.
Thank you very much, Morgan. Now up is Heather.
It's hard to pick just one thing. I think if I could do anything, there's a lot of talk about development of standard methods. For one, we're a long way from having enough standard methods that are true standard methods to have it really be meaningful for this industry. So one that's a long-term goal. And I also don't believe necessarily in the use of requiring a specific method for testing. Not every method is going to be fit for the variety of matrices we've already talked about. So I have concerns with that a method that claims to be good for, an edible product type is not going to actually be good for all the edibles. As an example, chocolate isn't always just chocolate, right? People might put mint in it or cinnamon or who knows what, and even those additives can affect the way a test performs. So I think really what I would like to see is much more robust and standardized method performance requirements, because in theory, any good method, as long as it meets the same performance requirements, should be able to produce results that can be compared. So I don't want to limit the methodologies. I want to see more robust expectations of the methods. And then just to totally throw something wild out there, part of me wants to revamp the entire system and require a much more, GMP type model for the industry. I still want to see some required testing to be verifying products, but risk-based assessment and risk-based testing of the products, because again, everybody's doing something unique in what they're making, how they're making it, and for a manufacturer to actually assess their process, and they have to do it the right way or this idea fails horribly, but if they can do a proper risk assessment and test in accordance with what they're doing, I think that the products would probably be a lot more consistent and more safe than they currently are.
Thank you, Heather. Okay, Chris, land your plane.
Coming in hot. I got to go back to micro. I think it's absolutely ridiculous that states have different approaches to the enumeration, what I would call quality indicator tests of total yeast and mold and total aerobic versus the pathogenic targets. You could see 100,000 CFUs per gram limit in one state, 1,000 CFUs in Illinois because they cited the American Herbal Pharmacopoeia's, monograph incorrectly, and then absolutely no requirements in states like Arizona and California. And I think that's really just, it doesn't make us look good, How do we come to such disparate interpretations of what's correct? I don't know. But I think harmonizing those, I personally would like to see total yeast and mold and total aerobic moved into a report only and addition of pathogenic targets, maybe on a risk-based approach depending on the count. But there's a natural microbiome in the plant, we all know that, and I really think that needs to be taken into account.
Thank you, Chris. Let's take it home, Darryll.
Okay, so I'll just repeat all the things I've said so far. I think we really need a harmonization of action limits, zero tolerance for high-risk pathogens, obviously, tiered action levels for indicator organisms, depending, again, on the matrix, the intended use, the route of administration, so that it reflects the real-world risk. Harmonization of method validation will help consistency and clarity and results for release. And then, like Chris said, adding some other organisms such as Pseudomonas to the profile that we test for. And yeah, that's it. So harmonization would help with all of these things and keep the states consistent. Thank you.
I'd like to thank the panelists for this dialogue, and at this time I'd like to open the floor for questions. So first, over here. Okay, let's see, who is this?
Hey, good morning, guys. My name is Ryan. I'm with Grow Defender. Great panel. One question I have for you. So, looking at this from the perspective like the med device space, everything is determined based off of risk. And Heather, I believe your last comment there, kind of moving more towards that. I'm curious to why maybe that's not been considered more before. And it seems like a lot of the issues could be resolved from just better sampling in general. And there's, standards like the ANSI Z1.9 standard that can, really make it easy to figure out how to sample different things. And I'm curious if you guys are actively recommending those types of standards for sampling. I was looking at the Colorado document that your sampling procedure, and I didn't really see anything about that. It's kind of, you guys are laying out what your recommendation is, but not really referencing a standard like the ANSI standard. And I'm just curious what the thought process is there, why you wouldn't recommend something that's been established and used in med and pharma and works really well and could potentially bring cohesiveness across the industry.
Great question. Complicated answer. So I know that our sampling scheme in terms of the sizes of the samples did look to ANSI standards in creating that. I just don't remember if it was 1.9 or specifically another ANSI document. Everything that we do goes through a public process, whether it's rulemaking, which is a formal public process, but in Colorado we also have something we call our Science and Policy Forum, where we gather public input on essentially anything that is a science-related policy decision or practice. And in that, it was actually through that forum of public engagement where our sampling guidelines were created. So part of it is that we do refer to standards when we think that they are great and that they apply easily and appropriately to what we're doing. But we also take that public input, and the public input at the time was to craft what we've crafted. And so that sometimes is why we're not always referencing a standard. There's other issues sometimes with referencing standards, is particularly within a regulation, or even sometimes if you put it in a guidance, right, then it's not enforceable. But it's also sort of sometimes then incorporating something as a requirement without actually making it a rule, which is not a great idea. So sometimes we have to be cautious with that. And some standards are not free. I think the ANSI one is, if I remember, but some standards aren't free, which is also then a challenge because we have to make everything that we incorporate by reference available to the user of the rule. So if we can't find a way to either get them free access, we can maintain a hard copy in our office and then we have to provide it. But that obviously isn't like a great way to make sure that the industry has access and knows how to comply. So there's just there's a variety of reasons in terms of what we do. We're also always reevaluating our processes and open to, again, if our document isn't great or it could be better, or we could incorporate aspects or just refer to a new standard when a new standard becomes available, we're always looking at that. So we're always evolving. But again, sometimes what exists today is just sort of the history of how we've got there. And then I was going to answer something about your other question, but I forgot it now. Apologies, you can ask again or come talk to me after.
Jeff.
Hey, great panel, guys. So I want to challenge you just a little bit because I feel like you've suggested a lot of great stuff, but I don't know if it's all available today. So I agree with you that it'd be great if we had standard methods for all of this. I agree with you that it would be great if we had sensitive molecular detection methods for every single pathogenic species and we knew the action limit that we needed to set and all that, right? But don't we not all have all that information yet? I mean, I don't think that the reason we have so many different regulations is because there's not a consensus about how to test cannabis for safety, right? So how far off is that and how do we get there?
I'll take a stab at this, Jeff. I mean, I want to use the common sense approach for a minute to total use in mold. How long have people been smoking cannabis? How many issues do we see? Is it really necessary to look at, even taking immunocompromised patients into account, how necessary is it to really put is 10,000 versus 100,000 CFUs per gram more appropriate? I don't know that we have the hard data, but we have decades, hundreds of years of using the plant to say otherwise. So I think part of this is we tend to want to put, as an analytical chemist myself, we tend to want to put data and numbers to everything to justify and prove and trust, and there's a lot to be said for that. But there's also, I think, the common sense side of things, which gets lost in translation sometimes.
I have one comment on this, and from an accreditation body standpoint, we cannot tell the laboratories what methods to use, and I think it is difficult for regulators to tell laboratories what methods they have to use. If and when they have tried that in the past, there has been significant pushback from the industry, and it's additionally very costly to implement a new standard method for the laboratories, especially if it's on different instrumentation. So if a laboratory has been running cannabinoids on HPLC for years and all of a sudden the regulator says, actually you need a GC for this method now moving forward, that is extremely costly for the laboratory, both in consumables and time, and they have to, of course, get it added to their scope, and there's all these, associated costs with it. So I think that part of the issue is with that pushback. I don't know the solution there because maybe Heather can speak to this, but from a regulatory perspective, I believe that regulators can be sued over things like this.
Yeah, I just want to add too, we often talk about making methods instrument agnostic, right? But a lot of the methods that are submitted are for specific instruments like the Agilent 6470 or a SciEx, and I couldn't reproduce that on some other instrumentation, right? So there's that issue where you basically, by requiring labs to use a specific method, sometimes you're requiring them to buy a specific piece of distribute their capital a certain way, and that's a consideration that, to Heather's point, is a serious one that has to be taken into account.
So, Josh Sweeter, I'm going to put my ACIL hat on. We produced a guide to homogenization of cannabis laboratory quality and testing standards 2 years ago. And this is getting revision right now and adding more to it. So I guess kind of the question I have is, do you guys believe that a it's a two-part question. Do you believe we should have a federal guideline or like a guideline where it's a state or a minimum, and then states, just like water testing or anything else, there could be a state-specific higher threshold or less threshold for contaminants, so states could still be able to do specific? That's question one. And question two is, the panelists, I hear, we should be doing this, creating national standards and everything. What are you guys individually doing to do so? It is a lot of time consumption to do these things. I mean, I spent 2 years on writing that guide with very minimal help. A lot of people in the industry want these things to happen. I'm just asking if you guys know any organizations or involved in any of this doing this standardization.
I'll take at least the first part, or say something about it. Anyway, I think a federal baseline would be lovely, right? Again, I mean, your states can expand upon regulations as they need to in their own state, assuming the feds allow that. But yeah, again, it would give us there's sort of that standardized baseline. It's complex, I think, and I think the feds have even acknowledged that, right? Because when we look at the products out there, we're talking about products that could easily fit into the category of a food, things that could fit into a dietary supplement category, things that are more like a nutraceutical. They're even, I don't know really if they're on the market, but I know people have wanted to make eye drops and they've wanted to make injectables, and we definitely have metered dose inhalers and things like that are out there, nasal sprays. So and we even have things that really are much more like medical devices and pharmaceuticals. So to craft one baseline standard is not easy to really make it successful, I think, at fitting all the products. But if we had standards from the feds that did fit these different types of products appropriately, I think that would be a really helpful baseline. Do I think we're going to have that anytime soon? Probably not.
Yeah, just to add on to that, I think I'm a huge proponent of that. It's kind of the environmental model. You got MRLs, and then states like California, for example, take that to another level. I think that makes a lot of sense. I think one challenge to that is understanding what's the minimum list that we need to look for, like 380 versus 10 in some states or none. What's the some states use different. I believe some states use different treatments, pesticides, depending on the climate or the regional pests that they have to deal with. So I think I could see a model where the federal baseline is that, and then states add on to that. I think your second question was about methods and what's being of the standard. I'm involved in the ACIL group. Yeah, shout out to Josh for leading that one. No, I mean, I think getting in front of people like at CANRA is very important. Visiting Washington D.C. for lobby days and getting on the Capitol Hill with staffers and having these conversations. I think a lot of the times, from our experience, is a lot of the people in D.C. want to do things and they just don't have the knowledge or the resources yet to actually implement change. So I think just, you mentioned case studies in the previous panel, Josh. I think case studies are huge. I think, politicians especially thrive on memos and case studies so they can point to their constituents and say, hey, this is why we need to do this thing. So I think, groups like ACIL, ASTM, really important in doing that.
Okay, we have time for just one question. Yes.
Good morning, everyone. I really enjoyed this panel. So I'm Dr. Krug from Bermuda, so from the Caribbean, and I'm a geneticist myself. I'm very new to medicinal cannabis, so I'm excited to be here. I had a comment actually on the previous question about regulating the standards of testing. I think there could be an opportunity to take from the clinical testing field, which is where I come from, because you've got labs doing, blood work on all different machines, but they just have a process where they get sent samples blindly and they have to make sure they get it correct. So that could be maybe something that can be used as an inspiration or a way to model after, because you've got labs all over the world doing blood tests and they get regulated. My question though is, has there been any conversation around these standards beyond the US from a global perspective? I know in the Caribbean, this is the biggest issue. Obviously, we grow cannabis very nicely, but the real issue with governments for passing laws on this is, well, how do we know it's safe what people are giving each other? So just wanted to know if there was any thoughts on the panel on bringing this conversation, or has there already been this conversation in a global context, and more specifically in the Caribbean?
I just wanted to comment on the provision of blind samples to laboratories. So I've always had the idea that the cannabis industry could potentially have something similar to WADA. So the World Anti-Doping Agency essentially has a double-blind external quality assessment scheme wherein they send samples to laboratories. Laboratories don't know that this is not a routine compliance sample. They think it's just a traditional sample mixed in with their batch. They give the results back. The results are analyzed and compared between all of the laboratories, and that result actually matters. So for a cannabis testing perspective, that would be incredible if there were an option for regulators to blindly distribute actual compliance samples, either retention from dispensary, previously failed products in some capacity, that they have characterized and homogenized, that they hopefully know the true value or an acceptable value for, distribute to all of the licensed laboratories in the state and compare the results. Now, some states do have a program that's similar to this already, and they're working on implementing it, but I am not aware of a state that has successfully implemented this in a double-blind capacity, which I think would be absolutely incredible for kind of catching issues where they are occurring.
Yeah, I agree. To your other question, I'm not aware of standardization efforts specific to the Caribbean or Caribbean countries participating specifically. I'm not saying they're not, I'm just not aware. But there are, again, a lot of international groups that are working on standardization, whether it's ASTM, AOAC is international, ACL is international? Yeah, ACIL. So again, a lot of international groups are working on this. CANRA actually has membership outside of the United States. Canada's a member, the Netherlands, I believe a country in Africa who I can't think of right now. It's only a few countries at this point, but it is open to international membership. So, I think there's a lot of international perspectives being considered in these different groups. Again, it's just sometimes it's challenging because states or even countries are limited by their own unique laws and their own unique administrative processes for either changing a law or changing a regulation. And again, that's part of the driver of why we see disparity between regulation. But I think, again, as standards that are international, that are really solid, that regulators in any jurisdiction can feel are appropriate and will actually be the solution to a particular issue, I think that will drive more standardization as those become available.
Okay, thank you very much. I'd like to thank the audience, all the participants for listening to the dialogue, asking questions. I'd like to thank the panelists for your time, participation, and I hope everyone learned something from this panel, and all for the sake of public health and consumer safety. Thank you very much, and I hope to see everyone in CanMed 26. Thank you.